Hello students, how are you? I hope you all are doing well at your home and you all are safe there and you all are taking all the precautions to avoid the spread of COVID-19. So today my class is on gene expression in E. coli. So this is very important topic of RDT. Yeah, today I will cover the mainly gene expression in Escherichia coli. So, what are the benefits of Escherichia coli for using it as a model for gene expression? Then, second is what are different elements of expression vectors? Then, what are different promoters? And one vector I will take as a model, that is PET vector, to explain the gene expression in Escherichia coli. Okay, so go ahead. Well, so today I will tell you why we have chosen this E. coli as a preferred host for the expression of heterologous proteins. The very first one is this is very simple in structure and its structure is well characterized, including its genomic structure. And there is very ease of working on this E. coli. This is the second benefit. The third one is E. coli has many vectors and strains available in the market commercially, and the one of the one most important uh, factor is that it is very easy to grow E. coli cells with less expensive media. Also, you can label any heterologous proteins with a lot of ease in E. coli models. So these were few benefits for using E. coli as a host system. Now there are certain drawbacks also of using E. coli, like as you all know, there is very less post-translational modifications like glycosylation, lipidation, proteolysis, etc. in E. coli model. So if you will express a eukaryotic gene in E. coli system, then there is chances of uh, production of inactive protein due to lack of post-translational modifications. Another drawback is in some cases there is very high rate of gene expression. So high rate of expression results in the aggregation of proteins, which leads to the insolubility of the proteins. Another drawback is there is lack of many chaperones. Chaperones are required for the folding of protein, so protein express may be misfolded. An expression vector has many elements like origin of replication. This is the site from where replication starts, and there is the selection marker like ampicillin resistance gene is here, and there is promoter T7 promoter is here. Promoter is the site where RNA polymerase binds. Next is ribosome binding site. This is the site where 16S rRNA subunit binds, and there is multiple cloning site where you clone foreign gene, and there is the transcription terminator site. This is for the termination of the transcription. There are certain regulator binding sites and other genes also. To understand how the genes expresses in E. coli, one need to understand what is promoter. Promoter is the region of DNA upstream to any gene. Plus 1 represents the first nucleotide of any ORF, and minus sign represents upstream sequences to the first ORF, first nucleotide of the ORF. Promoter is made up of two consensus sequences. One is at minus 35 position and another is at minus 10 positions. And these two consensus sequences are separated by approximately 17 base pair nucleotide sequences. So this is the promoter. Now the RNA polymerase binds over this promoter and progress in the downstream direction for the synthesis of mRNA. Another important structure is RBS, that is ribosome binding site. Basically RBS is found on mRNA. and this is required for the translation. In fact, this is the site where 16S rRNA subunit binds and it starts adding amino acids for the production of any protein. Hello students, so now I will tell you about the promoters in E. coli. There are many vectors available for expression of heterologous gene in E. coli system. These vectors all are under different promoters. They have different promoters. The commonly used promoter in E. coli is lac promoter. This is taken from lac operon. This is induced by IPTG or allolactose, and this promoter is regulated by lac O operator sequence and lac I repressor protein. This is promoter of high level of expression. This is a strong promoter, and the promoter is TRP promoter. This is taken from a gene of tryptophan biosynthetic metabolic pathway. This is also a strong promoter, and this is induced upon starvation of tryptophan or by addition of indole to acrylic acid. There are certain hybrid promoters also like TAC and TRC. These two are the hybrid promoters of LAC and TRP. They basically they are made for very high level of gene expression. For example, these hybrid vectors allows um, the accumulation of protein up to 15 to 30% of the total cell protein. Now, what is their structure? Uh, these both of them are made up of minus 35 region of TRP and minus 10 region of LAC promoter. What is different in the two? TRC has one additional nucleotide pair between minus 35 and minus 10 consensus sequence. So TRC promoter has a distance of 17 base pair between two consensus region, and this is used in PMAL vector, PTRC, PJAX vectors, and the regulation is similar to that of LAC, that is they are induced by IPTG. T7 is a very important promoter of E. coli expression system. This is basically promoter of gene 1 of T7 bacteriophage. This T7 promoter selectively binds with T7 RNA polymerase only. It synthesizes RNA at a rate several times of that of E. coli RNA polymerase. So this is very important promoter for very high end, very high level of gene expression. T7 RNA polymerase is resistant to antibiotics such as rifampicin that inhibits E. coli RNA polymerase. Another feature is T7 RNA polymerase can be inhibited by T7 lysozyme. So T7 is a very good promoter for the expression of heterologous gene up to about 40 to 50% of total cell protein. This promoter is basically linked with the LAC operon system, and T7 LAC promoters are being used in commercially available PET vectors and many other vectors. So this having linked with LAC system of LAC promoter system is now inducible by IPTG, and this is maybe regulated by LAC operator, LAC repressor proteins. To understand the expression of heterologous gene in E. coli system, I have chosen PET vector system. PET vector is very useful vector. This has T7 promoter. This vector was developed by Studier and colleagues of Novagen. So there are different elements of the vector, as it is supposed to be an expression vector like antibiotic resistance gene, origin of replication, promoter, T7 promoter, T7 terminator, operator sequence, at the 3' end of the T7 promoter, then ribosome binding site, then multiple cloning site. Description is given here. The important thing is to tell is this T7 promoter is followed by leg operator, where repressor protein coming from leg I binds and it stops the further transcription. But as the inducer is present inside the cell, inside the host, for example IPTG is present, then this IPTG will bind with repressor and this will derepress the operator. Similarly, derepress the promoter T7. And now RNA polymerase, T7 RNA polymerase will bind with the T7 promoter and it will start the expression of its downstream foreign gene. So this is the basic mechanism how the regulation takes place. So with the mechanism you must have understood different components also which are found in a PET vector. And now other than this, different variants are available in the market for each series. And for example here PET 11 A, B, C and D are available, and these differences are there in the reading frame only. You can see here they contain same restriction sites but differ in reading frames. So this difference is for reading frame only. These vectors, PET series of vectors may contain different tags like T7 tag, GST tag, HIS tag and many more. So this is the basic structure of any PET vector. I have just taken PET 11 as an example. The PET system involves not only an expression vector but also a genetically modified bacteria. The E. coli strain BLD3 is used for the expression of heterologous genes using PET system. What is modified in this host? Here is a T7 RNA polymerase gene under a lag promoter. And this host also has a gene for lag repressor, that is lag I. Lag operator is also found downstream to the lag promoter. So now you can control the expression of T7 RNA polymerase. As the inducer IPTG would be added, so this will start the production of T7 RNA polymerase, and this T7 RNA polymerase will bind with the T7 promoter of the vector and it will start the expression of heterologous gene. This is an overall preview of gene expression by PET vectors inside the host cell. So it is obvious here that uh, IPTG induction is de-repressing the expression of T7 gene 1 resulting in T7 RNA polymerase, and on vector side this IPTG induction is de-repressing the expression of target gene. Look here, this T7 RNA polymerase will bind with T7 RNA promoter in the absence of repressor protein upon induction by IPTG. So this is how a heterologous gene is expressed in using PET vector. Dear students, hope you have understood how the gene expression takes place in E. coli cells, and you have also understood how the PET vector works, what are different elements of the PET vector. In my next PPT lecture, I would explain the factors required for optimal expression of heterologous gene. Thank you. Your queries are welcome um, on my email ID or by any mean that you want to adopt. Thank you.